All right, so you're welcome to this live class on MCB 200. That's General Microbiology, Introduction to Microbiology. So basically, we are going to be focusing on major topics that are frequently asked, of course, frequently asked areas or frequently focused areas in exams. Okay, so basically, I want to actually have your comments. Okay, so I would actually understand when there is a network disturbance. Okay, so if you can hear me now, please just type I can hear you so we can commence immediately. All right, the previous life that I started had network disturbances, and because of that, I had to start this one again. All right, but remember we had two other classes on this course. All right, remember we had two other classes on this course that we actually focused on key areas. We looked at frequently asked questions and major topics that are usually very commonly focused on in the course. Okay, so we are going to be commencing with key areas, crucial areas today, and these areas are viruses, staining, microbial culturing. All right, so like I said, if you are not part of the previous classes that we had, the videos are still on this channel. Subscribe to this channel. If you haven't subscribed, then go to the live videos and check for the part 1 and part 2 of this particular course. All right, so we are going to be kicking off with viruses. All right, we are going to be kicking off with viruses, and we are going to be looking at viruses in terms of exam focus. All right, we are going to be looking at staining as well, and we'll look at microbial culturing. Okay, we are going to be looking at them in details. So this is of course a structure of virus. This is a virus. This is the structure of virus. So viruses are called acellular infectious agents. So they are acellular what infectious agents. Okay, they are called acellular infectious agents. What does it mean to be acellular? It's one C, then E, then double LULAR. They are cellular infectious agents, meaning that they are not made up of cells. That is, they don't have cells. All right, they can only replicate inside a living host cell. Okay, they can replicate inside a living host cell. That is the cell of the host where they have infected, the cell of the host when they have infected. So they can replicate, they can be formed from a living host cell. So they don't have cells of their own. That's one thing. So because of that, they are called acellular. So they are cellular infectious agents, which means that they can be able to transmit infection. They can be able to cause infections. All right, they are agents of infection. So they are called acellular infectious agent. That's what viruses are called. All right, so that's it basically. Now so basically, like I said, viruses replicate. When we say replicate, viruses forms inside the the cell of the host. The host is where they are actually inhabited. The host is where they actually live. All right, where they actually live. All right, so this is the structure of the virus. So quickly, what I'm going to be doing is I'm going to be explaining this structure. All right, I'm going to be explaining this structure. I'm going to be explaining the parts, all right, of this structure, and not just the parts of the structure. I'm also going to be explaining the functions of each of the parts, more like the roles that they play. All right, so if you can hear me, say I can hear you clearly. I want to be sure that I'm very audible. Say I can hear you clearly. That's if you can hear me. I need your responses right away. I can hear you clearly. That's if you can hear me. If you can hear me, say I can hear you clearly. All right, good. One response, another response. I need another response. I need two more responses. then we'll proceed. I need 2 more responses, then we'll proceed. All right, good one. Okay. All right, so like I said, this is the structure of the virus. This virus structure. All right, virus structure. So I'm going to be labeling it and explaining each part. Now this part you see inside here, this part you see inside here that is like this part is called the genome viral genome. All right, that's called the viral genome. All right, so what's the function of the viral genome? First of all, as you can see, the viral genome is located at the very center of the virus. All right, so this viral genome is called the genetic material. This viral genome is the genetic material of the what of the virus. All right, it's genetic material of the virus, and this genetic material can either be DNA or RNA. It can either be DNA or RNA. It can be either of these two. All right, so you know that usually anything that carries genetic material has to be the nucleus, right? And remember, it doesn't have a cell, so we cannot really say that it has a nucleus, because cells commonly have nucleus, apart from the ones that do not have defined nucleus. Those are prokaryotes. All right, the viruses do not have cells at all. They're not made up of cells. Remember, I told you that they replicate in the cell of their host. All right, so this is called the viral genome. So what they do is that they actually contain genetic material, which is the DNA or RNA. And what they do is that what this DNA and RNA do is that they actually make instructions. All right, they actually contain instructions that produce new virus particles. All right, remember I told you that they replicate themselves. So it's the presence of this viral genome that enables this virus to successfully replicate itself. So we are saying that it contains instructions. It contains instructions, and these instructions that it contains makes it actually easy for it to replicate itself. All right, these instructions that it contains makes it easy for it to replicate itself, makes it easy for new virus particles to actually be produced. So these instructions leads to the formation of new virus particles. Formation of new virus particles. So that's something you must take note of. All right, so that's the viral genome. Okay, so it's very crucial to note that. Very crucial to note that. Now moving forward, I've talked about the viral genome. The next one is called the nucleocapsid. All right, you see this one that accommodates the viral genome. This one here. All right, this one is called the nucleocapsid. All right, nucleocapsid. So it is more like the outer coverage. All right, it's more like the outer coverage. It's called a protein shell. So this is called a protein shell. Now is the protein shell, and it's actually, the protein shell is called the capsid. Okay, but because it is actually accommodating the viral genome, that's why it's called the nucleocapsid. So this one, the outer coverage is just like the nucleus that has inside of it DNA and RNA. Well, we don't call this one nucleus because this is not a cell. So what we call it is what? The nucleocapsid. All right, so it surrounds and protects the viral genome. Okay, so the combination of the viral genome and the capsid, which is the protein shell, this shell is called the capsid. So the fact that the viral genome is inside, that's why it's called the nucleocapsid. If viral genome is not inside, it will just be capsid. All right, if viral genome is not inside, it's just a capsid. And the capsid, like I said, is what? The capsid is what? A protein what? A protein shell. It's a protein shell. All right, so you must take note of that. So this is called the nucleocapsid. Okay, then we also have what we call the viral tegument. Viral tegument. You see these guys here, these guys that are outside here. Okay, you see here, you see these structures you're seeing here. This structure you're seeing here is called the viral tegument. The viral tegument. All right, viral tegument. So what is this viral tegument? This viral tegument is actually a cluster of proteins that occupies the space between the nucleocapsid and the outer envelope. All right, so they occupy this space. This space, this is the nucleocapsid, right? This is the nucleocapsid that we said. All right, then this is actually the envelope. Here is the envelope. This outer part is the envelope. This part you're seeing here is the envelope. Then this is what I labeled. These guys here is what I call the viral tegument. So they are all of them. All these things here are proteins. Remember, I told you that this nucleocapsid here is actually a protein shell. It's actually a protein shell. Then this is the viral tegument. So it occupies the space that is between the coverage, that is this nucleocapsid, okay, and this outer part. This outer part, and this outer part is the outer envelope. So what does this this structure actually helps? So there are proteins that help the virus to actually initiate infection once it enters the cell. All right, so once this virus you're seeing enters the cell, this viral tegument is what helps the virus to initiate, to start spreading the infection. All right, the viral tegument is, they are clusters. They are called clusters of proteins. Clusters of proteins, or protein clusters. So once they enter the cell, they help the virus to actually initiate the infection. All right, to initiate the infection. Then the envelope is the next thing. That is this guy here. Okay, let me use this pen. So this is the envelope. This is the envelope. All right, this is the envelope. So this envelope here, like that you're seeing, this envelope here that you're seeing, which is just after the what? Just after the what? Viral tegument. This is the envelope. So it's just like a cell wall or cell membrane. But because this is not a cell now, we cannot say that it is a cell wall or cell membrane. So it's actually made up of lipids. So, all right, so it's made up of lipids and acts as a protective barrier, just like we have the cell wall and the cell membrane that protects the cell. So it contains lipids, and we say it acts as a what? Protective what? Act as a protective barrier. So it acts as a protective barrier. Just like the cell membrane has, remember what cell membrane has what we call the phospholipid bilayer. All right, that this for cell membrane. No, then the cell wall has the cellulose. All right, these are the major substances or major features of the cell membrane and the cell wall. But in this case now, we have just lipids here. That is present in the envelope, this outer envelope. All right, this outer envelope. So it's called the protective barrier of the virus. So it's what protects the internal parts, the internal section of the virus. Remember what I said about viral tegument? It's viral tegument that helps the virus to actually initiate the infection. Remember what I said about the nucleocapsid? The nucleocapsid is actually the outer coverage, or it's like a protein structure or protein shell. That is capsid alone, no protein shell. The nucleocapsid, because it has what? Genetic material. it contains genetic material, it has genetic information. All right, it contains genetic material, that genetic information. So it's called the nucleocapsid. Then the one inside is what we call the viral genome. Is the genetic material itself, which can either be DNA or RNA. All right. Okay. So now, then the last one we have to talk about is this guy here. This guy you're seeing here. This one here. This guy is here. All right. These ones, they are called the envelope protein. All right. They are called. This is called the envelope protein. Remember, it's this one that is the envelope, but this one, all these structures you're seeing here, they are called the envelope proteins, or they are also called spikes. They are also called spikes. All right. So this protein structure you're seeing here, okay, sticks out of the envelope. If you look at it, it's connected to the envelope, so it usually sticks out of the envelope. So they act like keys. They act like keys that allows the virus to recognize and attach itself to specific host cells. All right. So what they do is that they act as keys. All right. These keys now enables this virus now to identify specific host cells. All right. Identify what? Specific what? Host cells. So it also identifies and recognize and also what? Attach. So you have to first of all identify, then you have to recognize that, see, this particular cell is the one that I'm supposed to bind to, or I'm supposed to attach myself to. Then after that, it will attach itself to it, and that must be a specific host cell, because the virus must attach itself to a host cell that is peculiar to it. The virus must attach to itself to a specific host cell. Just like you cannot use a key to open all the doors. All right. Every lock has its own key. You can remember in general biochemistry, the lock and key model of enzyme. The enzyme will only bind to its own active site, a specific active site, and if the active site is not the one that the enzyme is supposed to bind to, there will be no biological response created. Okay. So what these envelope proteins actually do, they are called spikes, is because they actually come out of this envelope. They are usually inside the envelope, so they project out. So once they project out, so the virus itself can be able to identify, recognize, and attach itself to specific host cells. All right. So these are the parts of a virus, and of course I've been able to explain all their respective functions. All their respective functions. All right. So that's one major thing that you need to know. All right. So these are the structural components. The structural components, number one, I talked about viral genome. After explaining viral genome, I went further to talk about the nucleocapsid. After the nucleocapsid, I talked about the viral tegument, which is this. After talking about the viral tegument, I talked about the envelope. After talking about the envelope, then I talked about the envelope protein. All right. So these are the major areas. Okay, these are the major areas of the virus, the structural components. These are all the components. Okay, now some key characteristics that you must take note of. Is that this virus do not have no cytoplasm. It doesn't have a cytoplasm. So there is no cytoplasm in virus. Another thing is that they do not have any ribosomes. No ribosomes. So because they don't have ribosomes, they cannot actually produce proteins. The proteins that are actually here are the ones that are in the structure already. They are the structural components. There are no additional proteins that are produced. Remember, I said that the capsid, all right, is a protein shell. I also mentioned that this envelope, these spikes here are envelope proteins. But there are no ribosomes that will synthesize more proteins for viruses. All right. Another thing is that viruses do not have any metabolism. So no metabolism as well. That is, they don't metabolize, which means they don't build up and they don't break down. Viruses are just how they are. Their major aim is to go to a particular cell and invade the cell, attach itself to the cell, and spread infections around that cell. All right, around that cell. So these are like key characteristics. And another thing is that they do know what, no cells, no cells. That's like a general characteristic, that they don't have cells. And because they don't have cells, that's why they don't have cytoplasm. That's why they don't have ribosomes. All right, because there are no what? Because there are no cells. Because there are no cells. All right. Okay. So now, how do viruses replicate? How do they replicate themselves? So we are going to be looking at viral replication now. Going to be looking at viral replication. Viral replication. I'm going to be explaining viral replication. All right. So viral replication actually happens through two major circles. Now these two circles are called the lytic circle, and also what we call the lysogenic circle. The lytic circle and the lysogenic circle, I'm going to be explaining. Now for lytic circle is when the virus attaches to the host. Now in lytic circle, the virus attaches to the host. All right. Then another thing is that after attaching it to the, it injects genetic material. That is the genetic material that we talked about now, that is DNA or RNA. Then after that, after injecting genetic material, now it now produces new viruses. This is the lytic circle. Then after after producing new viruses, what happens to the cell? The cell bursts. That is what we call lysis. The cell will just burst, like tear. It will just get destroyed, because once it has injected new material, this new material is what will be responsible for the production of the new virus. All right. So virus will attach to the host. It will inject. It would after injecting, it would what? After injecting, it will produce new virus. After producing new virus, the cell will burst. All right. The cell will burst again. When the cell bursts, the virus will attach again. After the virus attach, it will inject, like that. So it's a circle. It's a continuous process. And as this thing is occurring, that's how the virus is spreading and spreading. So that's for the lytic circle. So it's when the cell are burst that you say, that particular cell has been infected. Now, then for lysogenic circle is slightly different. Okay. Now, the viral DNA, let me just state it. Viral DNA integrates into host DNA. Remember that the first step here is that the virus attaches to the host, right, and injects genetic material. But here now, the DNA, okay, of the virus, that is the viral DNA, integrates into the host DNA. More like it now combines with the host DNA, and it will remains dormant. When after that, it will remains dormant. It means that it will not be doing anything. Then it's just like when an enemy comes in contact with more like a prey. Okay, the enemy of someone comes to someone you feel that is your friend, will just come and will just stay with you for a while. The person will not initiate any bad attitude, though, will not initiate any toxic traits. The person will just be there. When you say remain dormant, is that it will not act like a virus. Virus normally is supposed to infect, right? It's supposed to invade and infect. But this lysogenic it will come and it will just integrate. My friend, how are you? My friend will just stay, and at that point it will not even show any sign of infection. Then it will remain dormant. Now the period that is remaining dormant, it will be studying the cell. It will be studying the cell. It will be getting acclimatized. It will be knowing how the cell operates. Then boom, it will just activate. Now it will later what? Activate. This is called the lysogenic cycle of viral replication. By the time it activates, this one it will even be worse than the lytic cycle, because the lytic cycle it does not come or pretend, it will just come, virus attached to the host. Oh yeah, I be virus, so bam, before you do it will inject the genetic material, then it will produce new virus. Everybody will know that virus has come to kill, to steal, and to destroy. All right, then before you know, the cell will burst. This one in the lytic cycle, it actually shows its mission clearly. But in lysogenic, the mission will not be clear. It will integrate into the host DNA and it will remain dormant. It will not show any trait of viral nature. Then at the point later on, after it has studied the cell effectively, then it will activate itself and it will cause the cell to be destroyed. This time around, the several cells might even be affected, not just one cell, several cells, because by the time it has now studied one cell, it will now see other cells of that particular host to actually be the same. So it's just like you coming to a compound and you understand how that compound operates, and you know that that compound has a similar compound that is exactly like that compound. Anything you manifest in that compound that you come into, when you go to the other compound that is exactly like that, if you manifest that thing, the same response may be what you would see, because they have similar traits. So the lysogenic cycle is actually slower than the lytic cycle, but the rate at which the virus is activated here is faster than here. All right, so that's one thing you need to take note of. All right, so some common exam questions that are usually asked with regards to this. Okay, some common exam questions usually asked. I'm going to now question number 1. Viruses are dash: A, facultative parasites, B, obligate intracellular parasites, C, free living organisms, then D, autotrophs. So viruses are they facultative parasites? When we say facultative parasites, we are talking about parasites that usually start by attaching themselves to a very large system, a very large area of the body, like more like a parasite that affects the entire digestive system at once. Or are they the second option, are they obligate intracellular parasite? Intracellular means that they live inside the cell. Obligate means that they actually attach themselves to the cell and reproduce. When we say obligate, we are talking about reproducing. Then intracellular, it means that they reproduce inside the cell. Are they parasites that reproduce inside the cell? Free living organisms, do they spread round the body? Is there everywhere that viruses can actually go to? Then are they autotrophs? Autotrophs are organisms that partake in autotrophic nutrition, self-feeding. Okay, so do they produce their own food? Do they exhibit their own nutrition? Now the answer is not A. Because parasites do not go to a system. Do you understand? Parasites do not go to a particular system. Parasites go to a specific host cell, and that specific host cell must be a host cell that virus rather. Okay, I'm talking about parasites in context of viruses. Now viruses, they are not facultative parasites, because viruses, they do not go to every like region, like a larger region. They don't go to a larger region. They must be able to identify a host cell. Now, it is not C because they are not free living. Viruses cannot just enter the body and start roaming about. Viruses cannot just be roaming about. Viruses don't do like that. Okay? Viruses are not free living. They are not autotrophs because they don't feed on their own. Do you understand? They depend on the host cells. So the answer is that they are obligate intracellular parasites. Why? Because they actually live inside a host cell to reproduce. That's why they are called obligate intracellular parasites. They live inside the host cell to reproduce, in order to reproduce. Okay, so if you check your past questions, you actually see this question. All right, you see this question. So the answer is B, obligate intracellular parasites. All right, then I have another question. Okay, question number 2. Viruses consists of dash. A, protein only. B, nucleic acid and protein. C, lipid and carbohydrates. D, cell wall and membrane. So now remember what I was talking about. Okay, when I was talking about the different parts, remember I told you about the envelope. I told you about the envelope protein. I also mentioned the nucleocapsid. Okay, I also mentioned the nucleocapsid. All right. So then I talked about the I think I said viral genome. Mentioned the viral genome as well. Okay, mentioned the viral genome as well. So these were the 4 parts, and the 5th one I also mentioned viral tegument. Yes, I mentioned viral tegument. Okay, viral tegument, the one that is a cluster of proteins. So basically, this viral tegument is protein. Okay, viral tegument is protein. Envelope protein is also protein. Envelope contains protein. So basically, the nucleocapsid contains the viral genome and the capsid. And what is the viral genome? The viral genome is nucleic acid, where you have RNA and DNA. Remember, right? So it cannot be protein only, because it's not only proteins that we have. It cannot be protein only. The answer is nucleic acid and proteins. It's not lipid and carbohydrates. It's not lipid and carbohydrate, because it's only the envelope, this envelope, that has lipids. All right, but it does not have carbohydrates. So if it were to be if maybe lipid were to be included with nucleic acid and protein, I would have said yes. But lipid is in the envelope. All right, cell wall, it does not have cell wall, it does not have cell membrane, because it's not even a cell. All right, so the answer is nucleic acid and protein. Okay, now another question. Viruses lack dash. Viruses lack dash. A, ribosomes. B, nucleic acid. C, amino acid. D, none of the above. All right, now it's not none of the above, because the answer is here. So none of the above is out. It's not amino acid, because they have proteins, and proteins contains amino acids. In fact, amino acids are the building blocks of proteins. So amino acids are present in, if there is protein, then there is amino acid. You cannot separate protein from amino acid, because proteins are bonds of connections of amino acids that are linked together by polypeptides. Okay, so amino acid is not the answer, because amino acid is in viruses. Now it cannot be nucleic acid as well, because nucleic acid is in viruses. That's why we have the RNA and the DNA. So the answer is what? Ribosomes. Remember, I talked about some things that are, I even said it. Look at it when I said no cytoplasm, no ribosomes, no metabolism, no cells. Okay, remember I said that clearly. Okay, so now moving forward, we also have another question. Now this is the last question will be treating. Okay, now damage caused by viruses in cells is known as dash. So the damage that is caused by viruses in cells is called a cytopathic effect. All right, cyto means cell, pathic means pathogen, and a virus is a pathogen actually. So cytopathic effect is actually the damage that is caused by viruses in cells. So it's called cytopathic effect. Okay, now we have also what we call clear zones that are formed by viruses. Okay, viruses cause a very clear zones. More like when we say clear zones, we are talking about areas that other substances cannot actually go to because they have already been infected. Now these clear zones are called the plagues. They are called plagues. All right, they are called plagues. Plaques, plagues. All right. Now, then entry of naked viruses is called endocytosis. Endocytosis. When naked viruses enters the host cell, is called endocytosis. All right, endocytosis. Now, the enzyme in RNA viruses, enzyme that is in RNA viruses is called reverse transcriptase. Reverse transcriptase. Reverse transcriptase. All right. So that's one thing you need to understand. Now, so that's basically a set of questions that are related to viruses here. Okay. So that is it. And if you have any question on viruses as well, you have any question you want to ask on viruses, you can ask it in the comment box. I'm looking at the comments now. I can just immediately see your question and I will answer it. All right, but we have to move forward. Remember, I said we have to treat viruses and we have to treat staining. We have been able to treat viruses and exam questions under viruses. Now let's go to staining. After staining, we'll just look at microbial culturing. All right, after staining. But like I said, if you have any question whatsoever, feel free to ask in the comment box. So I want to know if you can still hear me. So okay, all right, okay, someone is asking what's the last one, please explain. When you mean last one, okay, I said reverse transcriptase. This reverse transcriptase is the genetic material in RNA viruses. Reverse transcriptase is called the genetic or the enzyme in RNA viruses. That is the enzyme that speeds up the reaction of RNA in a virus is called reverse transcriptase. All right, reverse transcriptase. So that's what I said. That's what I said. Okay, so moving forward, we'll be looking at staining in microbiology. Staining in microbiology. Please, you can put this down. Reverse transcriptase. That's what I said. Okay, reverse transcriptase. All right. So we'll be looking at staining in microbiology. Okay, let me get a fresh sheet. So we have staining. All right, we have staining. So we have couple of questions too that are related to this, but I have to explain what staining means first. So staining is actually the process of applying dyes to microorganisms. So it's the process of applying dyes to microorganisms. Okay, to microorganisms. Okay. Now it's not as though the adding dyes to microorganisms is irrelevant. If it was not relevant, we wouldn't have actually said that staining is a part of microbiology. Okay, but there are reasons why dyes are actually added to microorganisms. All right, so I'm going to be telling you these reasons. Now, the first reason why you stain is to actually enhance visibility. Enhance visibility. Do you know the reason why you're enhancing visibility? So that when the microorganisms are being viewed under the microscope, you'll be able to visualize them properly. Like they'll be able to be clear. They'll be able to be clear under the microscope when you place them on the slides to view. All right. Then another thing is that it helps to differentiate structures. It helps to differentiate structures. So the reason why you stain microorganisms is to differentiate structures. So you'll be able to actually use a particular maybe color of dye. You stain a particular structure differently from another, so you can be able to differentiate one structure from another. All right. Then another reason for staining is to identify types of bacteria. Okay, is to identify types of bacteria. So these are the three major reasons why we stain. Remember, I told you that staining is the process of applying dyes to microorganisms to enhance visibility, to differentiate structures, and to identify types of what bacteria. So one thing you need to understand is that most microorganisms are actually transparent. They're actually transparent. So because they are transparent, it will be difficult for you to actually see them under the microscope. So when you stain them, it will be very easy to observe them under the microscope. So it is very essential. Staining is not a minor practice in microbiology. It's a major practice. All right. So what is the, like I said, the purpose of staining is to enhance visibility. That is more like to increase the contrast. All right, to be able to, when you're identifying these types of bacteria, you're identifying the shape and arrangement. All right, so identifying the types of bacteria is actually to identify the shape and arrangement of the bacteria or the microorganism. It may not just be bacteria. All right, it may not just be bacteria. So you can be able to differentiate the gram, there's what we call gram positive, there's what we call gram positive bacteria. I've explained that in the previous live class. Gram positive and gram negative. Gram negative. Okay. Remember, I told you that gram positive stains purple. This one stains pink or red. Okay. So when you stain, you'll be able to differentiate gram positive from gram negative. Then you will also be able to, through this thing, you'll be able to also identify, as you identify the types of bacteria, you'll be able to also identify their internal structures. All right, the internal structures, like we have the spores, we have the what we call the capsules, and we also have, remember I told you about the flagella, which actually aids motility. All right, which actually aids motility. So when you do these staining procedures, you are going a long way. It's very, very important. It makes it easier for you. All right, so that's it basically. So now let's look at types of stains. Let's look at types of stains. All right, so we have types of stains. The first type of stain is what we call the simple staining. All right, we have what we call the simple staining. Okay, so I'm going to be explaining the simple staining for us. So one thing about the simple staining is that in simple staining you are just using one dye. Use one dye only. All right, and it shows the shape and the arrangement. Just shape and arrangement. You don't use more than one dye to do simple staining. All right. Then we have the common example of simple stains. We have what we call the methylene blue. All right, methylene blue. All right, so this is methylene blue. Methylene blue has a very sharp contrast, and has a very sharp response to transparency. In fact, if you use methylene blue to stain any microorganism that is very transparent. It acts very fast because it has affinity for transparency. All right. Then we also have what we call the crystal violet. Crystal violet. All right. Crystal violet. So this is another type of simple stain. All right. So we have the methylene blue and we have the crystal violet. All right. Then number two is what we call the differential staining. All right. It's all called the differential staining. Okay. So differential staining uses two or more stains. So this one uses two or more stains. This one uses two or more stains to differentiate bacteria. All right, to differentiate bacteria. So it's very, very important to note. All right. So one thing that is very common, I want you to note. One thing that is very common, I want you to note. Commonly asked question is that, although I've said it before, that staining was actually gram staining was actually developed by the scientist Hans Christian Gram. He was actually the one that developed the gram staining. All right. So the gram staining steps has to do with the use of the crystal violet as the primary stain. This is like the basic stain to use. Then after that, you use iodine. Then after that, you use alcohol as well. Then there's what we call counter stain. This counter stain will then counteract any form of stain that wants to spread so much around the bacteria. It's more like it will control the spread of the stain. So this one is called safranin. So it's called a what counter stain. It's called a counter stain. Then what is alcohol used for? Alcohol is used as a decolorizer. So alcohol, when you have used excess stain, it's just like when you have used a pen or a pencil to draw a particular thing and you want to erase. Then what do you use alcohol to do? Alcohol is to actually remove the color. Okay, is to remove the color. If the color is excessive, you use alcohol as a decolorizer. I believe you understand. All right, so you use alcohol as a decolorizer. That's what you do. All right. So it's very, very important to take note of that. Then we also have, still on the types of staining, we also have what we call spatial staining. All right, spatial staining. Number three, spatial staining. So spatial staining is actually used to detect specific structures. So it's used to detect specific structures. There are some structures that are very hidden. All right, that are very hidden, that are not very clear, that are not very visible. So when you do spatial staining, those hidden structures can be made visible. All right, so it's used to detect specific structures. All right, so these types of stains, we have what we call the capsule stain. Capsule stain. We have what we call the spore stain, and we have what we call the flagella stain. Now the capsule stain is used to detect the structure of the capsule. The spore stain is used to detect the structure of the spore, while the flagella stain is used to detect the structure of the flagella. All right, so like I said, these are spatial stains: the capsule stain, the spore stain, and the flagella stain. All right, so that's number 3. Now number 4, we have what we call negative staining. Negative staining. So negative staining is when only the background is stained. Only the background is stained. Only the background of the microorganism is stained. More like the upper surface is stained. The cell remains clear. Okay, the cell itself remains clear, or we can use the word transparent. Okay, so this one is actually used to detect delicate structures. When we say delicate structures, we are talking about those structures in the microorganism that can easily get damaged if malhandled. All right, you're trying to detect a particular structure. If you use staining for everywhere as well as the cell, you may destroy the cell. All right, you may destroy the cell. So that's something that is the basic thing. So now these are like the basic facts you need to understand about staining. You need to understand that staining is when you use attach dyes to microorganisms for the purpose of either enhancing visibility, differentiating structures, or identifying types of bacteria. Okay, then the types of the identification is knowing the shape and the arrangement. Okay, knowing differentiating gram positive from gram negative, identifying spores, capsules and flagella. Types of stains: we have the simple staining, which is used with only one dye. Differential, 2 or more stains. All right, spatial staining, which is used to detect specific structures. The negative staining, which is used to detect delicate structures. All right, so this is very very important for you to understand, for you to know. So any question under this staining that you come across, all right, do well to actually answer those questions. All right, so now we don't have all the time, we don't have much time. Let's look at culturing of microorganisms. Let's look at culturing of microorganisms. All right, so we'll be going straight to culturing. Like I said, after this class, do well to watch previous videos. Those two previous, the part 1 and the part 2 of this course. All right, no, I said gram positive is for gram positive gives purple. All right, while gram negative gives either pink or red. Okay, so we'll look at culturing of microorganisms. Okay, what I said about negative staining is that for negative staining, the background is only the background that is stained. More like only the, for instance now, if this is a cell, only the top aspect, the top areas will be stained. The internal area, the cell itself, only the outer part, the cell inside the cell will still be clear. Everything inside the cell, including the cytoplasm, the Golgi apparatus, the rough and smooth endoplasmic reticulum, even the cell organelle, all the cell organelles will still be clear. Only the background, only the topmost area, okay, only the topmost area that will be stained. Now the reason is that you don't want to damage the cell. You don't want to damage the cell, so you will not apply stain, because if you damage the cell, it will be difficult for you to actually identify delicate structures. All right, delicate structures. Okay, so now let's move to structuring of microorganisms, culturing of microorganisms. So culturing is actually the process of growing microorganisms in artificial media under controlled conditions. So it's the process of growing microorganisms in artificial media under what we call controlled conditions. Under controlled conditions. Now what is the purpose of culturing? Okay, there are purposes of culturing. Purpose of culturing, number 1 purpose is to isolate microorganisms. Now the next is to study characteristics. All right, to study characteristics of microorganisms. Now third is to identify pathogens. identify pathogens. Now remember, pathogens are agents, are disease causative agents. All right, disease causative agents, that is organisms that causes diseases. Now to also produce industrial products. So these are like the major purposes why we what there are major reasons why we culture microorganisms: to isolate microorganisms, to study microorganisms, to identify and to produce industrial products. All right, so please take note of this. So now remember, we said that it is the process of growing microorganisms, right, in an artificial media. Okay, that artificial media is like more like a cabinet, or more like a surrounding that grows microorganisms without going through the natural process of growth. All right, artificial media under controlled conditions. So what are these controlled conditions? Now these controlled conditions, I'm going to list some of them. Control conditions are proper temperature, suitable pH. Now every microorganism has a particular pH, okay, that they need in order to grow artificially. Then nutrients, there are specific nutrients as well. Then we also have oxygen. It's very, very important. It's a very, very important culture condition. So it can either be aerobic or anaerobic. All right. Then we also have sterile environment. Sterile environment is also required, because if the environment is not sterile, it can actually retard the growth. Okay, it can actually retard the growth of the microorganism. So now these are some of the conditions that are controlled conditions that are required for microorganisms to actually grow. Now let's look at types of cultures. All right, types of cultures. All right, a layman would think that these cultures have to do with the way people's day-to-day attitudes, people's way of life, and all of that. It's not that. We are talking about culturing bacteria. So number one type of culture is called pure culture. So what does pure culture mean? The pure culture is a culturing that contains only one organism, contains only one type of organism, only one type of microorganism. Okay, only one type of microorganism. This is the pure culture. All right, this is the pure culture. Number two is what we call mixed culture. So mixed culture, this one contains more than one type of organism. So contains more than one type of microorganism. All right. Then we also have what we call the arsenic culture. We have the arsenic culture. What does arsenic culture mean? It's completely free. This one is a culture that is completely free from contamination. That is, the microorganisms cannot be contaminated. Whether it be one type of microorganism or more than one type, the most important feature of an arsenic culture is that it is free from contamination. All right. Then the last but not the least, number 4, is what we call the enriched culture. So this one contains what we call added nutrients. To support growth. That's what we call enriched. So in this culture, okay, different types of varieties of nutrients are actually added, so the growth can actually be faster. So you can actually support the growth. All right, it can actually support the growth. So that is the enriched culture. Okay, so these are the types of culture. We have the pure, we have the mixed, we have the arsenic, and we have the enriched culture. All right, then now we'll be looking at culture techniques. All right, looking at culture techniques, more like some techniques are actually involved in culturing microorganisms. So I'm going to give you three major culture techniques. Number one is called the strict plate method. Strict plate method. Now, what is the plate method used for? So it's actually a technique that is used to obtain pure colonies. Pure colonies, more like when we say pure colonies, you are talking about more like a group of microorganisms in which all the microorganisms are one type. All right, are one type. So it's used to, all right, so it's used to obtain pure colonies. That's the function of the strict plate method. All right, then the second method I'm going to be teaching you is called the pour plate method. The pour plate method. Remember, the strict plate method is used to obtain pure colonies. Now the pour plate method is used to mix to mix mix microorganisms, used to mix microorganisms with others. So you're mixing microorganisms, okay? You're mixing microorganisms with other microorganisms. So it's used to mix microorganisms with others. And another thing is that in this pour plate method, so microorganisms can actually grow inside and outside. Okay, inside and outside. That is, when I say outside, I'm talking about on the surface. All right, on the surface. And now the surface, what you're actually using is what we call the agar. So the agar is actually a substance that is used during the pour plate method. So you mix microorganisms with others, and these microorganisms you're mixing, you mix it on an agar, and this agar is called molten agar. It's very thick. It's more like like a jelly-like. It's like a jelly-like agar. It's thick, but it's not like solid. It's like a semi-solid. So microorganisms can actually grow inside or on the surface of this molten agar. So that's what we call the pour plate method. You mix microorganisms with other microorganisms in an agar, and the microorganisms can either grow inside the agar or grow on the surface of the agar. All right, on the surface. Like I said, it's actually a substance that is used during the pour plate method. All right, it's not only the pour plate method as well. You can also use it in the strict plate method. All right, the strict plate method in this case, okay, when you're using agar in the strict plate method, what happens is that the bacteria gets diluted. Okay, it dilutes the bacteria, more like it reduces the concentration of the bacteria to a diluted form. Okay, that's what happens. That's what happens on the agar surface, because it's on the agar surface that you culture, whether strict plate, pour plate, or the third one is what we call the I think that should be the spread plate. Yes, spread plate. So spread plate method. All right, spread plate method. So in this case now, the sample will just be spread. This one is that it will be spread across the surface, on the surface of the agar. This one is nothing concerns it with growing inside. It's just spread on the surface of the agar. So this one spreads on the agar surface. It spreads on the agar surface. Okay, so that's how the sample of microorganism grows. It spreads on the agar surface. In the pour plate, it either grows inside or on the surface. But for the strict plate, okay, it's only used to obtain pure colonies. That is, you use it to actually collect, and after collecting, the bacteria gets diluted across the agar surface. Okay, across the agar surface. What did I say about pure colonies? I said pure colonies are like groups of microorganisms that are of the same type. All right, that are of the same type. So that is it. So these are the three culture techniques: the strict plate method, the pour plate method, and the spread plate method. Okay, all right, so it's very important to take note of that. Now quickly, the last but not the least thing we are going to be looking at is what we call culture media. All right, is what is called culture media. Culture media. All right, culture media. Now, what is culture media? Culture media are actually nutrient substances used to grow microorganisms. They are nutrient substances, okay, that are used to grow microorganisms in the laboratory. In the laboratory. All right. So they are nutrient substances that are used to grow microorganisms in the laboratory. Okay. So I want you to take note of that. So these are the core aspects of the definition of culture media. They are nutrient substances that are used to grow microorganisms in the laboratory. All right, in the laboratory. So what are the types of culture media? What are the types of culture media? So the types of culture media has a classification. Is that okay? Has a classification. Number one type of culture media based on classification. One classification is based on physical states. Based on the physical state of the culture media, we have different types. We have the solid media. I'm going to be explaining each of them. We also have the liquid media. And we have the semi-solid media. So we have the semi-solid media. All right. So based on the physical state, we have the solid media, we have the liquid media, and we have the semi-solid media. So now, what is the solid media? This solid media contains agar, and it's used to grow colonies. More like used to grow group of or collection of microorganisms. So the solid media contains agar and is used to grow colonies. So example of solid media. We have what we call nutrient agar. So nutrient agar is an example of solid media. Majorly it contains agar and it's used to grow colonies. All right. Then that's for solid media. Then now the second one here is called the liquid media. Now liquid media is also known as broth. Okay, it's also known as broth. So in the liquid media there is no agar. All right, there is no agar, and it is used for what we call bulk growth. All right, more like growth of microorganisms in a bulk. So it doesn't really have to do with colonial or the growth of colonies. It has to do with bulk growth. All right, bulk growth, like in bulk, like in very large amount. Okay, in very large amount. So bulk growth. All right, that's for liquid media. So an example of liquid media is what we call the nutrient broth. All right, the nutrient broth. Okay, nutrient broth. Okay, so now for semi-solid media has what we call low agar concentration. Okay, it's called low agar concentration. That is a media that has low agar. The concentration is low, so it's almost dilute. So it's used for what we call motility tests. All right, so it's used to test the movement of microorganisms. All right, it has agar, but the concentration of the agar is very low. All right, in the semi-solid media. Remember, this is based on the physical states. All right, this is based on the physical state. So that is it. So it's a major thing you need to take note of. All right, so an example of this semi-solid media is what we call molten agar. I've talked about molten agar before. All right, so molten agar is actually it's semi-solid. Remember, I talked about molten agar when I was talking about the culture techniques. In fact, when I was talking about that pour plate, the pour plate method, I said it's molten agar that it's usually there. The colonies actually grow inside and on the surface of molten agar when they've actually mixed together. All right. So that's something you need to take note of. So these are the types of culture media based on the physical state. All right. Then now number two is what we call, another classification is based on the chemical composition. Based on the chemical composition. So based on the chemical composition, we have what we call defined media. Defined media. All right. We also have what we call complex media. So these are the types of media when you are classifying it based on the chemical composition. All right. So defined media is actually a media in which the exact chemical composition is known. All right. The exact chemical composition is known. That is, you know the exact chemical composition of the microorganisms, all right, in the culture media. Okay, remember that the culture media contains nutrient substances that grows microorganisms in the laboratory, that used to grow microorganisms in the laboratory. So when you know the exact chemical composition, all right, that is what we call the defined media. That is what is called the defined media. All right. Then we have the complex media. This one is for defined media. Then for complex media is when the chemical composition, exact chemical composition is unknown. All right, is unknown. Okay, it's unknown. So this is for complex media. Why this one is for defined media. All right. So when the exact chemical composition is known, that is defined. When it is unknown, that is complex. All right. Then number three is based on function. Based on function. All right. Based on function. This is the third and last classification. Under based on function, we have selective media. All right. We also have what we call differential media. I'm going to be explaining each of them, and I'll also give examples. All right, we have differential media. Then the third, the last but not the least, is called the enriched media. I talked about enriched media before now. All right, so we have the selective media, we have the differential media, and we have the enriched media. So what's the selective media? So this selective media is actually a media that allows the growth of specific, we are talking about the functional. So functionally, selective media allows the growth of specific what? Microorganisms only. Microorganisms. That's why it's called selective. Because it has the exact microorganisms that it wants to grow. So that's why it's called the selective what? Media. All right, that's why it's called the selective media. So example of this selective media, common example is what we call the McConkey agar. So that's what it's called, the McConkey agar. All right, so this one, what it does is that it selects only gram negative bacteria. The McConkey agar selects only gram negative bacteria. That is one thing that is common about McConkey agar. So don't forget that selective media allows only the growth of specific microorganisms. Then now let's talk about differential media. All right. So when we talk about differential media, one thing that is common about differential media is that it differentiates organisms based on appearance. So it differentiates organisms based on appearance. So organisms that have the same appearance, it groups them. Like this organism have a particular appearance, this microorganism have a particular appearance, it stays here. The other organism have the particular appearance, it stays here. So it differentiates organisms based on appearance. So a common example is called the blood agar. Common example is called the blood agar. That is an example of differential media. Then now for enriched media contains extra nutrients. Okay, enriched media contains extra nutrients. It contains extra nutrients. That's why it's called enriched. Contains extra nutrients. Okay, it has lots of nutrients compared to other media. This one is that it even helps organisms to even grow faster, and it helps organisms to actually grow better. It helps organisms to actually grow better because of the maximum level of nutrients that it has. All right. So an example of this is what we call the nutrient agar. Nutrient agar. That's example of the enriched media. It's called the nutrient agar. For differential, I told you blood agar. For selective, I said McConkey agar. All right. So these are the three different classes or classifications of culture media. I say based on physical state, we have solid, we have liquid, and we have semi-solid. I say based on chemical composition, we have defined and we have complex. Based on function, we have selective, we have differential, and we have enriched. All right, don't forget that. Okay, so it's at this point that we would come to the end of this class. All right, we've been able to treat viruses, we've been able to treat staining, and we've also been able to treat microbial culturing. All right, so I would like you to answer any question you come across based on what you have learned tonight. Also, try as much as possible to watch the part 1 and part 2 of the general microbiology classes that we had previously, and I wish you all the best in your exams. Okay, so this is where we draw the curtain for tonight. All right, all the best. Don't stop studying, don't stop reading, don't stop learning, don't stop growing. See you at the top. Goodbye.